So my name's James Lee. I am a clinician scientist group leader here at the Crick. And I want to just for a second just debunk exactly what that means. In really simple terms, what it means is I actually have two jobs. The first job is I run a research group here at the Crick. And the Crick is an amazing building. You've all sort of seen it as you came in today. But actually, the thing that makes this a real privilege is the opportunity to work with a really spectacular group of young scientists. And these are the people shown on the slides. Some of them are in the audience today to embarrass them. But they are amazing. And actually, they're the real heroes in this piece because they're the people who've actually done a lot of the research that I will be talking to you about today. My other job is actually up at the hospital. So I'm a consultant gastroenterologist. I work up at the Royal Free Hospital, and there I run a clinic where we look after patients with inflammatory bowel disease. And some of those are the local patients from the local area here, but actually there's an awful lot of patients who get referred to us from elsewhere in the country because they have particularly difficult or complex disease and they want our opinion in terms of how to treat it. So what actually is inflammatory bowel disease? So it's a term that you may have heard of, but actually it's not a single thing. It's actually an umbrella term that captures two different diseases. The first is called Crohn's disease. This was a disease that's actually named after an American pathologist called Crohn, who first described this in the 1930s. Actually, interestingly, it had been described by a Scottish pathologist about 20 years earlier, who many of the Scots will feel slightly aggrieved that it was never named after their guy. The other disease is ulcerative colitis. Now, these two diseases are lumped together under the umbrella inflammatory bowel disease because they share many common features. They're both lifelong diseases. They're both incurable diseases. They both have a relapsing, remitting course, so they have flare-ups and then times when the disease gets better. They're both driven by the immune system. So actually what essentially happens is your immune system gets its wires crossed. So instead of fighting off coughs and colds and keeping you nice and fit and healthy, they start attacking the bowel. And that attack on the bowel causes inflammation. It can cause ulceration. It actually then eventually can lead to damage. And the complications of that damage actually often require surgery. So Crohn's disease can cause narrowings in the bowel, strictures as we call them, and they can cause blockages. Or it can also cause times where the inflammation actually burrows its way out of the bowel and actually into organs nearby, and we call those fistulas. Ulcerative colitis can present in a very severe way and not respond to any of the treatments we have, and then you end up having to do an emergency operation to actually take someone's bowel out. And if you don't do that, people can die. And actually, prior to the advent of treatments, there was a significant mortality associated with inflammatory bowel disease. But these are two different diseases, and I want to give you a very sort of whistle-stop tour about the key differences. So Crohn's disease can affect any part of your digestive tract, from your mouth all the way through to your bottom. And in fact, it's patchy. So you can have bits of bowel that are affected by Crohn's and then intervening bits of bowel that are entirely normal. The inflammation in Crohn's also spans the entire width of the bowel wall. So ulcerative colitis is actually different on all of those 3 points. It's not patchy, it's usually continuous. It begins in the rectum and then works its way proximally. Some people it doesn't go very far. Some people it just affects the rectum. In other people it can affect the entire of the large bowel, but it doesn't go outside the large bowel. That's why it's called a colitis. It only affects the colon. It also doesn't affect the full thickness of the bowel wall. It just affects the lining. And that's why it gives you slightly different symptoms to what we see in Crohn's disease. Now, if we think about the sort of constellation of symptoms that patients get with inflammatory bowel disease, it can be many different things. But actually, we often see certain patterns of symptoms that go along with one disease or other. So in Crohn's disease, it's characterized by abdominal pain. This is a symptom most patients will get at some point. It also is characterized by diarrhea. Now, when we say diarrhea, we don't just mean everyone in this room will have had diarrhea at some stage. It's not just the fact that the stool is a bit looser. It's the fact that when you've got to go, you've got to go. And you've got to run to the loo. And sometimes people don't even get there in time. Crohn's disease can also cause mouth ulcers. It can cause abscesses or infections around the bottom. Ulcerative colitis, on the other hand, is slightly different. It typically presents with rectal bleeding and diarrhea. And often the diarrhea starts first, and when it gets bad, you then start noticing that you're passing blood. And during a bad flare-up, people can be going to the loo 15 or 20 times a day, to pass blood mixed in with their stool. Now, those are the symptoms that's specific to those two diseases, but actually there's many of the symptoms that are actually common to both. Both can cause weight loss, unintended weight loss. This isn't the sort of weight loss that you're aiming for when you go on your diet. This is the sort of weight drops off you and you have no way of controlling that. It can cause profound fatigue. You know, I've heard in past talks people refer to this as tiredness, and patients get really annoyed when you just refer to this as tiredness. This isn't tiredness. This is debilitating fatigue. This is sleeping 20 hours a day, waking up and still feeling tired at the end of that time. And actually testament to the fact that these are systemic diseases. They're not just intestinal. They actually can cause inflammation in other parts of the body as well. So you can have joint pains and arthritis. You can have eye inflammation. You can actually have skin inflammation, all of which can go with these diseases. Now, it's really important to say that this is different to irritable bowel syndrome. Now, irritable bowel syndrome is really common. Most people in this audience will have had symptoms of IBS at some point in their lives. The key difference, even though it causes some of the same symptoms, is that irritable bowel syndrome does not cause inflammation, and it does not cause damage, and it is not life-threatening. So if I do a colonoscopy, which I often do when I'm up at the Royal Free, if I see a picture of the colon inside and it looks like this, then I'm really happy, because this is an entirely healthy colon. You can see the background mucosa is nice and pale, and you can see this sort of lacy network of blood vessels. This is exactly as you want it to look. Unfortunately, sometimes when I'm doing colonoscopies, I don't see that, and I see this. And this is ulcerative colitis. So you can see immediately that what you have here is redness, inflammation. You have this sort of exudate, and actually this mucosa is really friable. It doesn't take much when you contact it for it to start bleeding. So I promise that's the last gory slide I will put up. So who gets IBD? Well, actually, the thing is, most chronic diseases, when you think about chronic diseases, you kind of expect them to be older people. But actually, that's not what IBD is. So inflammatory bowel disease typically presents somewhere between the ages of 16 and 30, maybe 16 and 40 to be a bit fairer. And think about everything that you really want to do between those ages. So you want to finish your education. You might want to go and get your degree. You might even want to be doing a PhD. You might want to get your first job. You might want to have a relationship, maybe get married, maybe even start a family. And IBD can actually cause problems that interfere with all of these activities of living that we all sort of kind of take for granted otherwise. So it can be really debilitating for patients. The other reason why IBD is really important is it's getting commoner. So over most of the 20th century, we were seeing this steady increase in the rate of both forms of IBD in the Western world. And by that, I mean North America, Western Europe, generally the Northern Hemisphere. But interestingly, over the last 30 years or so, we've started to see increases in IBD in countries that we'd never previously seen it in before. So we're now seeing it in the Middle East, we're seeing it in the Far East, we're seeing it in sub-Saharan Africa. And the one thing that seems to link the emergence of not just IBD, but actually lots of autoimmune diseases, is a westernized diet, or westernization. And that's not to say it's all due to the diet, because there's lots of things that change as our societies become more westernized. But what that means altogether is that we are seeing this inexorable increase in the prevalence of inflammatory bowel disease. Now, this is global data, but actually this is UK data. So this is nice data from Scotland, where they were slightly worried, looking at the rates of increase, that by 2028, we would be dealing with double the number of patients that we were dealing with just 20 years ago. And the point at which we got to that point was when the prevalence would hit about 1%. Well, unfortunately, the prevalence has now hit 1%, so it's happened earlier than we expected. So we can say that today, in 2024, as a gastroenterologist who looks after patients with IBD, I'm dealing with about twice the number of patients that I was looking after when I qualified as a doctor 16 years ago. So the chances are that there's more than 100 people in this room. The chances are there's going to be someone here who has IBD. If there's not someone here, there's almost certainly you will know someone who has IBD, even if you don't know it, or even they don't know it, maybe. So the question then is, how do we treat these diseases? And that's complicated. And it's complicated because for a long time we've recognised that some patients get IBD and actually have a very mild disease course. They don't flare up very often. They can be treated usually with the first-line treatments we have. but some patients get a really aggressive form of IBD that need much more in the way of treatment. So historically, we've always had this as the way in which we would do our treatment. So this is the sort of step up. So you start at the bottom with the mildest drugs. They're also probably the least effective drugs. If those don't work, you then go up a level and you go to something a bit stronger. And if that doesn't work, you go up a level again to something stronger still. And you keep walking up this staircase until you eventually find a drug that actually works. Now that ensures that we will never overtreat people. We're never going to give you drugs that you don't need. But actually it also means that if you're that unfortunate patient who really needs the strongest drugs at the top, you're going to have an awful long time trying other things that are ultimately not going to work for you. So actually recently we've changed the way we start to think about how we treat that. And instead of going up, we start to think about coming down. And what that means is we then start with the most effective therapies at the beginning in the hope that we can then switch off the disease, really effectively. And part of the reason for that was actually a study that we published earlier this year. And this was a trial, and nested within this trial, we were taking people newly diagnosed with Crohn's disease, and they were either getting the standard care that we currently give, or they were getting the best therapies given at the beginning. And actually what we showed was that if you give people the best therapies early on, they're doing much better. So there's a much lower chance of them needing an operation or needing courses of steroids, which we know over time actually have lots of side effects. There's also a much higher chance of you actually getting healing in the bowel. So we want to see a bowel that looks like that first picture, and that's achievable with the right therapy. It takes a bit of time, but you can get to the stage where you no longer see inflammation and ulceration. Now, the reason we didn't do this earlier was there was always a concern that if you give people lots of immunosuppressants, well, you're going to expose them to risks and potential side effects of those drugs. Except weirdly, you don't. Because actually the data from our trial showed that even in the people who got 2 immunosuppressants right at the beginning, there was no increased rate of serious infections. That's weird, isn't it? We're giving people immunosuppressants, but they're not getting sick with infections. Part of the reason for this is actually the thing that really predisposes to people is uncontrolled IBD. And actually, if you really get them well, actually you don't have this increase in infection rate. But whenever you think about risk, you have to think about both sides of the equation. What we found was that actually by giving people early effective therapy, we reduced their chance of needing operations quite dramatically. So 1 in 20 of the people who were managed with the standard way ended up needing an operation within their first year because of complications of their Crohn's disease. Whereas only 1 person in the whole of the 190 odd in the top-down arm needed an operation. And actually that wasn't really even an operation for Crohn's. So if we're going to give, advanced therapies, what are the advanced therapies? Now, not that long ago, it was really easy to be a gastroenterologist because we didn't have to make any decisions because we only had 1 class of drug. But actually now what's happened is we've now got lots of different classes of drug and often lots of different agents within those classes. So, it's now much harder to manage IBD because we have to start making some decisions. But even with all that, we know that we still have to call our surgical colleagues for help to actually do operations to take out bits of diseased bowel in about 1 in 10 people with ulcerative colitis within the first 10 years they're diagnosed, and maybe 2 in 10 patients with Crohn's disease. Now, those numbers are slightly lower than they used to be. It used to be quoted it would be 20% in ulcerative colitis and maybe 50% in Crohn's disease, but they're not 0. And the reason they're not 0 is this. So despite all those great drugs and all the great options that I have to choose from as a gastroenterologist, in the back of my mind, I always remember this. So this is data from ulcerative colitis. And on the left-hand side of the dotted line are the remission rates for all of the drugs in their pivotal trials that ended up getting them approved. That means you got the drug and within the first 8 or 12 weeks, your disease went into remission. On the right-hand side is the rates of remission with the placebo. And so even though we have lots of drugs, I hope you can see that actually these drugs are only working really well and really effectively at an early time point in somewhere between 15 and 40% of our patients. So I've talked to you a lot about what IBD is, but many of you will be wondering, well, why do some people get it and other people don't get it? So IBD is complicated. And it's not as straightforward as saying we can predict who will get it. In fact, what the sort of traditional dogma is that you need some genetic risk. You then need to see an environmental trigger that actually sets the whole process off. And what it fundamentally boils down to is your immune system attacking your gut because it perceives there's a problem there when actually there isn't. And that's often because it's misidentified bugs that live within your gut as being something that need to be cleared and damaged when in fact they're not. They're the friendly bacteria we would normally hope to have. So when my patients ask me why and how I got IBD, the analogy I like to use is the firing of a gun. So genetics kind of loads the bullets into the gun, and then the environmental trigger pulls the trigger, and that's what then causes it to fire. So if you imagine, if you have a gun that's got no bullets in it because you have no genetic risk whatsoever, it doesn't matter if someone pulls the trigger, it's not going to go off. But equally, if you have a gun that's fully loaded with bullets and you never see the environmental trigger to pull the trigger, it's also not going to go off. So you need to have both things. And just like the firing of a gun, once IBD has developed, there's no way to unfire it. There's currently no way to cure that disease. So let me go through a few of the bits, and this is going to be very high level and pretty whistle-stop, and you're welcome to ask questions about this afterwards. So let's think about the contribution of the immune system. Well, again, if I said IBD as a whole was complicated, the immune system bit is even more complicated. And that's because there's lots of different cell types in our immune systems, and many of them have been implicated in certain ways and shapes and forms in the development of IBD. To just give you a sort of high-level summary, what's generally thought is that there is some damage to the lining of the gut, the intestinal epithelium. That then allows the bugs that live within our gut to be exposed to the immune system that sits within our gut. There's more immune cells in your gut than in pretty much any other organ in your body. What that happens is that triggers a sort of damage signal saying something's gone wrong, the bugs are here, they shouldn't be here, quick, come and help and get rid of them. And one of the first things that happens is you get this huge influx of inflammatory monocytes and macrophages that are designed to produce chemicals to try and get rid of those pathogens. They're also to try to sort of gobble them up. That in turn enables the T cells. You will have heard about T cells from COVID. There's been a lot of sort of publicity about T cells. But the macrophages and then dendritic cells then activate the T cells, and then you get this second wave of inflammation that in turn then causes more damage. And all of this occurs when the normal checkpoints that are within our gut fail to stop it. So we have cells within our gut that are designed to suppress these sort of abnormal immune responses, and they don't do it the way they should do. But that is a massive oversimplification of what we understand about IBD. And there's lots of other cell types that are important. I don't want to diminish really exciting research that's being done around the UK and internationally on some of these other cell types. So let's think about the genetic component. Now, normally, when you hear about a disease that's genetic, you think, well, if I've got it, then one of my parents probably had it, and my children will get it, and my brothers and sisters will get it. But actually, that's not the way in which the genetics works in IBD. It's more complicated than that. You can see a theme emerging here. In fact, the reason we know that genetics is important in IBD, we've known it for over 35 years, and we've actually known it from really elegant twin studies. So these are studies that were done in the late 80s, and they took sets of twins. So as you know, you can have identical twins who share exactly the same DNA, and then you can have non-identical twins who have only 50% of the same DNA. And what they did was they looked to see where one twin had inflammatory bowel disease, what was the chance of the other twin having inflammatory bowel disease. And of course, the nice thing about twin studies is twins grow up in the same environment, so they're likely to have seen all the same sort of environmental triggers if it's in a westernized diet or whatever it might be. And what they found was that if you had identical twins and one had IBD, there was about a 60% chance that the other would have IBD. If you had non-identical twins, that chance was only about 5%. And it's kind of true to say, so a lot of my patients, when I talk to them about this, the first thing they ask is, well, are my kids going to get this? And actually, the answer to that is almost certainly no. If you have inflammatory bowel disease, the chance of any of your first-degree relatives getting inflammatory bowel disease in their lifetime is somewhere between about 5 and 10%. So there's a 90% chance they won't get it. But then the background risk in the population is less than 1%. So it's much higher than it would be if you had no family history. And actually, when I'm seeing a patient, sometimes I always ask them, is anyone in your family had problems like this before? It's the commonest and it's probably the strongest indicator that someone will have IBD if they have a strong family history of it. And in fact, much of the last 35 years or so, since these twin studies, has been spent trying to find what are the genes that are involved in the development of IBD. And again, it's not just one gene, it's likely to be multiple genes acting together. And actually, that's been a really successful enterprise. So we now know of over 400 regions in our DNA that are directly involved in developing IBD. Now that's great, and it means we've kind of solved the where. We know where things are going on, and we know that that is involved in these diseases. What it hasn't solved is the how. So we don't know how most of these changes in our DNA contribute to getting these diseases. And I'll come back to that in a minute. So then what's the environmental trigger? Okay, you can't change your genetic risk, but what's the environmental trigger that actually sets this off in the first place? Well, again, remember, these increases that we've seen over the last 100 years or so can't be due to genetics because the genetics hasn't changed in that time frame. It must be due to some sort of environmental trigger setting this off. And again, just like everything else in IBD, it's a bit complicated because we know that there's lots of things. It's not as simple as saying it's a Western diet. Because is it the diet, is it ultra-processed food, for example, or is it the fact that we now consume a lot more emulsifiers than we used to? Is it the fact that we now have microplastics in our diet? Is it the fact that actually we use a lot more antibiotics in a Western lifestyle? Is it the fact that we have higher rates of obesity and stress and pollution and all these other things? And the answer is that what might be the cause for one person is almost certainly not the cause for someone else. So there is a lot of research going on into what the environmental triggers are. Most of them are difficult to modify. And actually, we still don't have a mechanistic understanding of how they initiate disease. So then, in the last part of this, I want to just talk to you about some of the work that we actually do here at the Crick. And in terms of thinking about the motivation to this, I want to take you back to this figure I showed you a few minutes ago. You'll remember that what this shows is that actually, for all the advanced therapies we currently have, most of our patients do not get better with most of the drugs we have. So some might work for one person, but then if it doesn't work, we may have to try something else and try something else. And if that sounds a lot like trial and error, well, that's kind of what it is. But it's even worse than that. Because actually, if you think about how these drugs are developed by pharmaceutical companies, what happens is they have to go through a whole series of trials. They start with phase 1. Those are safety trials, make sure they don't do anything horrible and nasty. Then it's phase 2 to find the right sort of doses we have to use for patients. And then it's phase 3 to actually then test to make sure the dose we've picked actually works. But if you take all of the drugs that are going into those trials, only about 1 in every 10 ends up being approved for the indication it was meant to be starting. So 90% of these actually fail. So why do they fail? Well, interestingly, the vast majority fail at what's called phase 2. And phase 2, the job of a phase 2 trial is to show that the drug actually works for people. And the most of them fail because the answer to that question, does this drug actually work? The answer is no. And what that means is it's not that that drug is not binding to the inflammatory chemical it's designed to bind to, or it's not inhibiting the enzyme that it was planned to inhibit. What it actually means is that doing that in people actually doesn't make any difference. It doesn't make them better from the disease they're suffering from. So it's kind of nuts, isn't it? We're investing so much money and so much effort to take drugs through, and fundamentally, we're targeting many of the wrong pathways. Because when you switch off the pathway, it does nothing to the disease process. So that brings me back to a sort of fundamental question, which is, how well do we actually understand IBD? Because I would argue that if we had a really, really good understanding of what's going on, we probably wouldn't be wasting money targeting the wrong pathways, and we'd probably have drugs that work in more than 15 to 20 to 30% of our patients. So if we accept that maybe, in spite of everything I've told you and all the complexities, maybe we still don't understand these diseases well enough, then actually the next logical question is, well, how can we understand IBD a bit better? And I'm biased because I'm a geneticist, but I would argue that actually genetics gives us a pretty unique opportunity to better understand why people get IBD. And that's because in many areas of science, you never quite know when you're studying a disease, if you find a change in someone's body, was it a cause of the disease? Or maybe it was caused by the disease. But with genetics, the genetics always came first. So it kind of resolves that cause-effect relationship in a way that's actually very helpful for us. The other reason why genetics is a huge opportunity is actually we now have a very detailed understanding of all the bits of our DNA that are contributing to these diseases. Remember, we know the where, but we don't know the how. And actually, there's even more evidence that genetics might be the right way to do this. Because if you take drugs that are going into those phase 1 trials, and you just select the ones that are targeting a pathway that has been implicated by disease genetics, then that drug has actually got a far higher chance of actually working. So that was originally estimated to be maybe a 2-fold chance. But actually, if you really understand the pathway in detail, it's probably about a 6-fold chance. So you can take your 10% success rate and you can turn it into something approaching 60% success rate. Of course, there still may be safety issues why some drugs don't work, but you should at least get beyond the sort of the efficacy hurdle. But there's a really big problem. Most of the findings where we're finding bits of our DNA that are linked to these diseases are not occurring in the actual protein coding sequence of genes. And what that means is they're not having predictable effects on protein structure or function. In fact, the vast majority lie in the slightly more enigmatic non-coding genome. And they're very strongly enriched within these weird things called enhancers. Now, enhancers are a bit like the volume dials in your genome. They turn up or turn down the amount of genes that are made nearby. And if you think about it, every cell in your body has the same DNA in it. But what makes a heart cell and a liver cell is what genes are turned on or off in those individual cell types. And the thing that controls that are enhancers. So enhancers are really important, but they're also complicated because an enhancer may be active in one cell type and not active in another. That's why it can turn on a gene in a liver cell and not in a heart cell. So if you want to figure out what's going on, you've first of all got to figure out, well, what cell type is this enhancer working in? And then you've got to figure out what gene is it regulating. And often the genes aren't right next door to it. And only when you figure that out can you then say, well, what's the genetics actually doing to this process that actually increases the chance of someone getting IBD? So what all of that means is that while genetics has this amazing potential to not only transform how we understand the disease, but also potentially how to treat it, much of that potential is currently unrealized. So where does that actually leave us? Well, it leaves us with very long lists of genetic associations. Now, some people have slightly unkindly said it's basically like reading a telephone directory, for those of you who remember what a telephone directory looks like. The problem is they're not actually that far off the truth. So what we have is we know where the association is, we know what chromosome it's on, we know where in the chromosome, we even sometimes know what the genetic variant is or variants that may be contributing to the disease at that site. But that's kind of where the sort of certainty of our knowledge ends. And in fact, the next column, and I know this because I was involved in writing these papers, is usually a geneticist's best guess as to what gene might be actually driving the disease there. And the problem is this is incredibly inaccurate. So look at this one here. We've named 2 genes. It's probably not both. It might be one or other. But then the question is, well, are there any other genes there? Well, yeah, there's another 63. How sure are we that it's definitely one of the 2 that we might have heard of before and hence we put in the paper, as opposed to any of the other 63? Well, the answer is not that confident. What about these regions? Here's something that's maybe even harder. Here you've got association and say, well, how many genes are actually within the region where we've got this strong genetic signal that's increasing your chance of getting IBD? Well, none. What do we then do? Well, the simple answer is, and what has been done up and for a long time, is people have kind of gone, let's ignore that. That's a little bit too tricky. We'll focus on the ones where we can kind of make a good story up. And we'll just, all of this other stuff, it's just too fiddly. So that's not the approach we take in my lab. So in my lab, we like to see all of this set up as a bit like a treasure map. Because actually, what we know is we know where there's something really important going on that directly impacts your chance of getting these diseases. And actually, if it's in a region where we don't know anything about it, that's great. Because actually, we want to go and dig for treasure. We want to go and actually figure out what is going on in the hope that maybe it will tell us something new that we didn't know previously about the disease. So while I'm a scientist and I run a lab, actually, I'm a treasure hunter. And what I like to do is think about how do we go and find the treasure that's lying within these bits of DNA that is telling us why these people get the diseases in the first place. So I want to tell you about one treasure hunting expedition that we went on recently and published a couple of months ago. So this is a region on chromosome 21. It's one of those regions that if we'd highlighted and said how many genes are there, the answer would have been 0. So it's what used to be called a gene desert. That's a slightly simplistic term, but it just basically means there's nothing there. And the really cool thing about this gene desert is it doesn't just increase your chance of getting both forms of inflammatory bowel disease, it actually increases your chance of getting a whole load of other inflammatory diseases as well, including ankylosing spondylitis, which is a nasty inflammatory spinal disease. It also increases your chance of getting primary sclerosing cholangitis. That's a very long name for an inflammatory liver disease for which actually we don't have any effective treatments at the moment. So we thought two things. First of all, if this is a region that increases your risk of at least 5 different diseases, it's got to be pretty important. And the second thing we thought was all of these diseases are immune mediated. They're all caused by your immune system getting its wires crossed and attacking those organs. And therefore, we thought this must be an enhancer, because enhancers often lie in these gene deserts, and it must be an enhancer that was switched on in an immune cell. So here is that region. You can see that at the peak at the top here, where you can see a couple of tiny red dots and the bit in blue, that is the association. And there's no genes immediately underneath it. There are a few genes nearby, but there's nothing immediately at that site. So we said, okay, let's go and dig for treasure. Let's go and see if we can find where there might be an enhancer switched on. And if there is an enhancer, what cell type is it switched on in? And the nice thing is we didn't have to think about every cell type in the body. We thought it must be immune, because these are all immune-mediated diseases. So we took all of the immune cells and we used a method that enabled us to see, is there an enhancer, one of these volume dials, is it switched on? And the answer was yes, but it was only an enhancer, it was only switched on in a type of immune cell called monocytes. And monocytes are the precursor for another type of immune cell called macrophages. So I'll use those two terms kind of interchangeably. So we think, okay, that's brilliant. We found that there's an important cell type. We know that actually monocytes and macrophages are implicated in these diseases. What else can we then learn? Well, the way I've put this DNA up on this slide, it looks like a long, nice straight line. But if you were to take a really, really big magnifying glass and go and look inside a nucleus of a cell, you'd see something that looked much more like a bowl of spaghetti. So in fact, it's not nice and straight. There's bits over here that are physically coming into contact with bits over here or vice versa. But all of this is regulated. And actually using another method, we were able to find out that the site of where the genetic association is, the site where the enhancer is physically touching a gene, not one of the two that were right next door that a lot of people thought might be the cause at this site, but actually one that was, a good few miles upstream. Obviously not miles, but in genomic terms, a good few miles upstream. And that was a gene called ETS2. And the other thing we found out was that if you carry the genetic variant that increases your risk of all these diseases, you had higher levels of this gene ETS2, specifically in your macrophages. And that's great. And that, you could almost have stopped at that point, but the obvious question that you will all ask is, well, what on earth is this gene and what's it doing in macrophages to increase your chance of getting all these diseases? Well, one of the nice things that we can do in the lab, if you imagine it's a situation whereby, imagine you're trying to figure out which fuse in your house controls your lights. How do we figure it out? Well, we go to the fuse box and one by one, we turn them all off. Well, actually, that's kind of how we figure out what genes do as well. So what we can do is actually using a technique called CRISPR, which you almost certainly have heard of, we can actually turn off genes. So we can make macrophages, inflammatory macrophages, in fact, that look just like the ones that come out of the gut of patients with IBD, and we can turn off ETS2 to see what's it doing. Well, remarkably, when we turn off ETS2 in macrophages, it switches off pretty much all of the functions that actually enable them to contribute to disease. So they lose the ability to migrate, so they can't actually get into tissues. They lose the ability to make pro-inflammatory cytokines. These are the inflammatory chemicals, many of which we target with our best therapies. They also lose the ability to make reactive oxygen species. These are the chemicals that actually cause the damage in the tissue. And they lose the ability to gobble up the bacteria and get rid of them. They lose the ability to phagocytose. So you've got 3 or 4 of the absolute core functions of an inflammatory macrophage, which seem to depend on a single gene. So if you could turn something off, actually the other thing that's quite nice to do is to then look at the effect of turning it on. This is the sort of, you walk into a room and you see a switch, what does the switch do? I don't know, let's turn it on, and let's find out what it does, and then the light appears at the back of the room. So what we can do is we can take resting macrophages that are not inflammatory at all, they're nice, they're calm, they're peaceful, and we can turn on the ETS2 gene in those cells. And we can do that actually using a method very similar to how they make your COVID vaccine. So instead of making a bit of RNA that encodes part of COVID, we make RNA that encodes the ETS2 gene, and we load it up in these cells. And then the question is, well, what does that do? Well, remarkably, turning on just this one gene turns on all of those pathways I told you about before. And actually, even more remarkably, if you look at the cells that you get at the end of that experiment, those cells look just like the cells that you get out of the gut of patients with Crohn's disease. So people have previously published a sort of signature of all the genes that are very highly expressed in these inflammatory disease-causing macrophages. And we can show that well over half of those genes are significantly turned on by just overexpressing one gene in a resting macrophage. And that's pretty remarkable, because you're basically saying that you can take a non-inflammatory resting cell, you can turn on a single gene, and it looks just like it's come out of the gut of someone with Crohn's disease. And actually, even more remarkably, pretty much all of the most effective therapies that we currently use target cytokines that are principally produced by macrophages. And all of those cytokines get turned on by turning on ETS2. So whether that's we're thinking about TNF or IL-23, which are probably the classes of drugs that are the most effective in IBD patients at the moment, both of those go on, but so do a lot of other inflammatory cytokines, many of which have been linked to resistance to either of those drugs. So when we do genetic studies, the goal of what we're doing is not just simply to stamp collect, to find 400 hits. The idea is that with enough hits, we can start to identify the genes, and then we hope that those genes will start to then coalesce into pathways. And that's the real gold when we're treasure hunting. That's the real thing we're really looking for, is to understand the pathway. Because while you can have many different genes, fundamentally you often end up with a sort of smaller number of pathways, and all of those pathways need to go wrong to get these diseases. So the real goal here is to find the pathways that underlie this. Now, one thing we did, having found this new pathway whereby macrophages can be turned into these inflammatory little monsters by ETS2, is we said, how enriched is that pathway compared to everything else we've previously think we've understood from IBD genetics? To try and understand, to put this in context, to try and say, how important is this relative to anything else? Well, it turned out, to our surprise, that this was more enriched for the genetics of IBD than anything that's ever been described before. So, IBD has been one of the sort of poster children of genetics, because we find pathways that have been implicated in things like Crohn's disease, one of which is called autophagy. And we found that autophagy was important in Crohn's disease because there were 6 autophagy genes that came up from our GWASs. When we looked at how many genes were in the ETS2 pathway, it was over 25. So we found, essentially, we've stumbled on, if you like, not only a sort of master regulator of inflammatory macrophages in humans, but actually we've stumbled on a pathway that appears to be one of the absolute core drivers of inflammatory bowel disease. And we've kind of known for some time that macrophage inflammation is important in IBD, not only because if you look in the gut of patients, there's tons of inflammatory macrophages everywhere, but as I've told you, it's because many of the cytokines, the inflammatory chemicals that these cells produce, are the ones that we target with some of our best therapies. Now, that might be targeting an anti-TNF, or it might be targeting an IL-23, but don't forget that targeting one of those chemicals often only makes 20 or 40% of people better. And sometimes, I regularly am in the experience of we try one drug and it doesn't work, so we have to try the next drug, and sometimes that doesn't work and we have to try the next drug as well. So the really exciting thing is that we think we found the thing that sits right at the top of the pyramid, that actually turns on these macrophages in the first place. And that if you imagine, if you could come up with a drug that could target that, then it doesn't matter whether you need a bit of an anti-TNF or a bit of an IL-23 or a bit of something else. You potentially have a way of turning down the whole pathway. So you may not be in the position of having a drug where it only works in a fraction of patients. You may have a drug that actually is much more effective. Now, some of you will have already read about this research. It was published just before the summer, and it received quite a lot of attention in the media and sort of beyond. And part of the reason it received a lot of attention is actually we have found a drug that does appear to be able to switch off this pathway. And that drug is something that's already licensed for use in people. It's used for other things like cancer and neurofibromatosis. It's not used for anything inflammatory. Now, that drug is not safe enough to be taken for long periods of time. And of course, IBD is a lifelong incurable disease. I told you that at the beginning. So actually now what we're doing in the lab is actually trying to find ways of making that much safer that would enable us to take it in a much longer form, in the hope that actually that could give you a much more effective treatment. So patients will not be having to sort of, play the Russian roulette of, will my drug work, won't my drug work, will I need something else, will that work or not? We actually will have a drug that potentially could actually turn off one of these central pathways that we know is driving IBD. So I want to come back to this because actually, as I said to you at the beginning, the real heroes of this story are the people in the lab who have done this work. So actually, this work was driven by two people in particular, Christina Stanke, who is a PhD student, who is sitting just over there, and Christoph Borge, who is a principal lab research scientist, who actually was my first person I ever employed in my lab when I started my lab in Cambridge about 10 years ago, and has foolishly followed me everywhere ever since. The work that we're now doing to actually turn this back to becoming a drug, because actually I'm not in this to publish great papers. I'm not in this to do media interviews. I'm fundamentally in this because I'm fed up of sitting in clinic trying to scratch my head and figure out why this drug hasn't worked for the patient and will the next drug work or not. What I'd like to do is for those patients who are trying to finish their education, trying to get married, have kids, get their families sorted out, is actually have a way in which we could offer them a drug that would be much more effective and would actually have a much higher chance of getting the disease under control so they can get back on with their lives. And Ashna, who's up on the top right, is the person in my lab now who's actually taking this project forward and actually working to develop the next generation of drugs built on genetics in order to hopefully have better treatments for people in the future. So science can't be done without funding, and these are just a few of the people who very generously funded the science that we do. That's pretty much all I wanted to talk to you about this morning. So if anyone has any questions, I'd be very happy to take them.